Hello students, in today's class we'll discuss the mechanism of DNA replication. In the last session we discussed Hershey and Chase experiment. Let us start with the mechanism of DNA replication. The two questions arises in our mind. One, what is DNA replication? When does it takes place? So let us find the answer for these questions. When it takes place. The first one, what is DNA replication? So the process of making exact or identical copy of DNA is called DNA replication. During the process of DNA replication, the two twisted strands separate and separated strands act as a parental strand, or we call it as a template strand on which new strand is synthesized. And the daughter strands are exactly identical to the parental strand. Okay. So during this process what happens? One of the parental strand is conserved and new strand is synthesized on the parental strand, so that hence the DNA replication is commonly known as semi-conservative method of replication. Semi-conservative method of replication. So the only DNA is capable of replication, whereas you remember one thing, RNA is synthesized from the DNA. RNA is synthesized from the DNA with help of enzyme called transcriptase enzyme, or we call it as a DNA dependent RNA polymerase enzyme. With the help of DNA dependent RNA polymerase enzyme, okay, RNA is synthesized from the DNA. If you want to synthesize DNA, which enzyme is required? Simple. Okay, we required DNA dependent DNA polymerase. If you want to synthesize DNA polymerase enzyme is required. If you want to synthesize RNA, DNA dependent RNA polymerase enzyme is required. So when it takes place, normally the DNA replication process takes place just before the cell division, and you already know that during cell division, okay, cell cycle, there are two phases. One is interphase and M phase, and during interphase, that is S phase of cell cycle, S phase of cell cycle, or S phase of interphase, the DNA replication process takes place. And you remember one thing, the process of semi-conservative replication is proved qualitatively by J. Herbert Taylor and quantitatively by Michelson and Stahl. Okay, so two things you remember. What is DNA replication? When it takes place. So the process of synthesis exact or identical copy of DNA is called DNA replication, and it takes place during S phase of cell cycle. Okay, S phase of cell cycle. So what are the requirement for DNA replication? So the first requirement is, okay, and before we go into the requirements, the mechanism of prokaryotes is better understood than the eukaryotes. So we learn mechanism in E. coli. Okay, requirements. The first requirement for DNA replication is the nucleotides, substrates, nucleotides. If you want to synthesize DNA, we require 4 nucleotides: deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate and deoxythymidine monophosphate. If you want to synthesize RNA, we required 4 nucleotides. Okay. Adenosine monophosphate, guanosine monophosphate, cytidine monophosphate and uridine monophosphate. So here we are doing DNA synthesis. We required 4 nucleotide: AMP, DAMP, DGMP, DCMP and DTMPs. The substrate molecules. And you remember, all these nucleotides of DNA are obtained from energy source, that is DATP, deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate and deoxythymidine triphosphate. And from all these triphosphate, 4 deoxyribonucleotides are obtained. So energy provided when 2 phosphate bonds are broken down. So for DNA replication we required energy, and that energy is ultimately come from triphosphates. Look at here. So when 2 phosphate bonds are broken down, the energy is used, and that energy is utilized for synthesis of DNA. And second requirement is a DNA template. I told you, when you look at the structure of DNA, double helical structure, double helix structure. At the time of replication, the 2 strands unwinds. The unwinded strands act as a template strand to synthesize a new strand. The separated single strand of double helix serve as a template strand or parental strand on which nucleotides are added or new strand is synthesized. The next one is primer. The question arises, what are these primer? A short segments of RNA or short strands of RNA nucleotides. So what is the significance? You remember, for DNA replication, primers are absolutely required. Without primer, we cannot synthesize DNA. And these short segments of RNA are formed at 5 prime 3 prime end of the prime direction. And these RNA fragments provide free hydroxylic group or free hydroxylic group end for addition of DNA nucleotides in 5 prime 3 prime direction, 5 prime 3 prime directions. Then set of enzymes. So along with the enzyme, we required one very important is inorganic ions, that is Mg+2. So the list of enzymes which are involved in DNA replication, the first one is DNA helicase enzyme. So what is the role of DNA helicase enzyme? DNA helicase enzyme breaks hydrogen bonds between 2 strands. Between 2 strands. Look at here. So helicase enzyme which breaks the hydrogen bonds. So these separated strands act as a template strand on which new strand is synthesized. The unwinding of double helix synthesis. Next one, DNA gyrase, or we call it as a topoisomerase. Look at here, what is the role? So when helicase enzyme breaks the hydrogen bonds, at the opposite end, creates a tension. That tension is released by DNA gyrase enzyme. DNA gyrase enzyme. Primase enzyme. So the primase, RNA primase enzyme, which helps in synthesis of RNA primers, short segments of RNA. So these short segment of RNA provides free hydroxylic group for addition of DNA nucleotides at 5 prime 3 prime direction. Then DNA polymerase enzyme. I told you for synthesis of DNA which enzyme is required? DNA dependent DNA polymerase enzyme. There are three polymerase enzyme which plays a very important role in the replication process: DNA polymerase 1, 2 and 3. The DNA polymerase 1, removal of RNA primers. So once for a synthesis of DNA, we required RNA primers. Once addition of DNA nucleotides takes place on the template strand, then no role of RNA primers, and those RNA primers are removed by DNA polymerase enzyme. So just imagine. So the RNA primers are present. Look at here. Sorry. So RNA primers are in between. Those RNA primers are removed by the DNA polymerase 1 enzyme. DNA polymerase 2, proofreading. Look at here. This is a parental strand. The nucleotides are guanine, cytosine, nitrogen bases. Guanine, cytosine, thymine and adenine. And you already know that. So during synthesis, exact copy of DNA is formed, and in between two strands you find a complementary base pairing. Look at here. Guanine base pairs with cytosine in the other strand. Then cytosine, guanine, cytosine, thymine, adenine, thymine. Look at here. So one at the time of addition, there is one nucleotide, wrong nucleotide is added to the other strand, daughter strand. So instead of thymine base pairs with adenine, instead of adenine you find thymine, and these DNA polymerase enzyme which correct the nucleotide sequence. DNA polymerase 3. So it helps in synthesis of DNA, or it adds nucleotides of on the parental strands. DNA ligase, it joins two DNA fragments. So RNA primers are removed by DNA polymerase enzyme, and the fragments of DNA are joined with the help of DNA ligase enzyme. So let us start with the mechanism of the DNA replication. The first step which is involved in the mechanism of replication is the activation of nucleotides. So nucleotides of DNA such as deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphates are activated and phosphorylated by deoxy ATP, that is deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, deoxythymidine triphosphate. Okay. So, and soon after the replication, the first enzyme, that is helicase enzyme, which breaks the hydrogen bonds between the two strands, and two strands unwinds. Okay, and this is we called as the initiation point, or we called as a origin of replication, or ori site and ori site. And in case of prokaryotes, prokaryotes have only or replication bubbles and have single origin of replication site, monoreplicon, whereas in case of eukaryotes, possesses many origin of replication site, and that we called as a multiple replication site, or multi-replicon. Then the unwinding of double helix, you already discussed. The unwinding of double helix occurs from the ori site, origin of replication site, and it is catalyzed by an enzyme called helicase enzyme. So unwinding provides a single stranded template for replication. So two separated strands act as a template strand. So look at here. A protein called helicase breaks the hydrogen bonds of the two strands of DNA and forming a Y-shaped structure, and that Y-shaped structure is called a replication fork. And after the formation of replication fork, the two strands are stabilized with the help of proteins. Okay, those are called binding protein. The binding protein keeps two strands apart, okay, apart, preserving the replication fork. Or these binding proteins which prevents recoiling of DNA separated two strands. Then soon after helicase enzyme, the topoisomerase enzyme, okay, relieves the stress at the opposite end of the DNA. Look at here. So what exactly? Because of supercoils, what happens? Okay, uh, it creates it resembles like a spring-like structure. If you remove the coil at one side, opposite strand tension is created, and that tension is released by topoisomerase enzyme. Topoisomerase enzyme. It relieves this coil. Okay, so look at here. So it relieves the supercoils. Supercoils. So that replication bubbles are formed. Soon after the replication bubbles, now nucleotides has to be added on the template strand. The problem is directly DNA nucleotides are not added to the template strand. In order to add DNA nucleotides, we required free hydroxylic group, and that free hydroxylic group is provided by the RNA primers, and those RNA primers are synthesized with the help of enzyme called RNA primase. The enzyme RNA primase makes a short segments of RNA at 5 prime 3 prime direction, 5 prime 3 prime direction on the template strand. So about RNA primers synthesized about 10 nucleotides of RNA, and these RNA provides 3 hydroxylic group to initiate DNA synthesis or the polymerization of DNA nucleotides that runs in 5 prime 3 prime direction. And you remember one thing, always you remember one thing, replication process always takes place on the template strand, always takes place in 5 prime 3 prime direction, not in the 3 prime 5 prime direction. It always takes place in 5 prime 3 prime direction. Then so addition of denucleotides, once the RNA primers are formed, hydroxyl group is available at 5 prime end, okay, of 5 prime directions. So immediately the free hydroxyl group helps in attachment of DNA nucleotides, and those DNA nucleotides are added by DNA polymerase enzyme. Nucleotides are added to exposed bases of parental strand. Addition starts from 3 hydroxyl group end of RNA primers. So this is a short segment of RNA, one end it has a free hydroxyl group. So free hydroxyl group immediately, so it starts at 5 prime 3 prime direction, 5 prime 3 prime direction, and that takes place with the help of DNA polymerase enzyme. Look at here. So two separated strand, one runs in 5 prime 3 prime direction, other runs in 3 prime 5 prime direction. I told you the replication always starts at 5 prime 3 prime direction. Look at here, DNA synthesis always proceeds in 5 prime 3 prime direction. Addition of nucleotides in one strand proceeds towards the replication fork continuously. Look at here, continuously nucleotides are added. Which nucleotides? DNA nucleotides are added towards the replication fork continuously, and that strand we called as a leading strand. Okay. Then in the opposite strand, okay, again the replication process takes place in again in 5 prime 3 prime direction. Here the addition of nucleotides are not continuous, they are discontinuous. A short segments of DNA are formed. Short segments of DNA are formed, and that strand we called as a lagging strand, and these fragments of DNA are called Okazaki fragments. They are called Okazaki fragments. So when these Okazaki fragments are formed, immediately those Okazaki fragments are joined with the help of DNA ligase enzyme. Look at here. Soon after the addition of DNA nucleotides, uh, we have to remove the primers and we have to fill the gap, and later those gaps are filled, and proofreading after proofreading the DNA fragments are joined with the help of DNA ligase. So removal of primer, gap filling, and ligation. Look at here, the primers are removed. Okay, the primers, these are the RNA primers, they are removed with the help of DNA polymerase 1 enzyme, which replaces it with a DNA. So DNA polymerase enzyme remove the RNA primers. Then Okazaki fragments are now joined by phosphodiester bond catalyzed by an enzyme called DNA ligase enzyme. DNA ligase enzyme. Then look at here. The formation of new, new DNA strand. An enzyme called DNA polymerase 3 adds DNA nucleotides. ಯಾವ ಡೈರೆಕ್ಷನ್ ಆ್ಯಡ್ ಮಾಡ್ತದ 5 ಪ್ರೈಮ್ 3 ಪ್ರೈಮ್ ಡೈರೆಕ್ಷನ್ ಆ್ಯಡ್ ಮಾಡ್ತದ ಟು ದಿ RNA primer on the template DNA. The continuation of DNA synthesis. DNA strand synthesis continues in 5 prime 3 prime direction with a new strand termed as a nascent strand, or we called as a leading strand ಅಂತ ಹೇಳ್ತೀವಿ. The proofreading checks DNA bases. DNA polymerase enzyme proceeds along with the nascent leading strand continuously, and proofreading DNA nucleotides replace the incorrect DNA sequences or DNA bases. The formation of continuous DNA. RNA primers are removed by proteins, okay, and the proteins called ligase, okay, that is DNA polymerase 1, and the proteins called ligase which joins DNA fragments two DNA fragments. At the end, termination signals, okay, termination signal is signaled by specific sequence of nucleotide on DNA, and that ends the replication process. Look at here, these are replication bubbles. So replication process takes place in both the direction. Okay, replication process takes place in both the direction, hence it is called a bidirectional and bidirectional. Proofreading. So sometime wrong nucleotides are added to the new strands. Look at here, DNA polymerase enzyme removed the incorrect nucleotides, adds the right one. These are the sequences, the earlier sequences. Look at the green color one. This is a capital, okay, in the parental strand we have a thymine, in the daughter strand we have again thymine. The thymine is replaced by, okay, adenine with the help of DNA polymerase 2 enzyme. Okay, so this completes the mechanism of DNA replication or semi-conservative method of DNA replication. In the next class or in the next session, we'll discuss experimental proof for semi-conservative method of DNA replication. Thank you.